So now we're going to talk a little bit about epidemiology. And let me say at the outset that in terms of my knowledge, I've had some nursing students in this program that are already registered nurses that know a lot more about epidemiology than I do because that's the field that they work in, which is great. Anytime you have, whether it's me or a different instructor, anytime you have additional information, that's an important part of this course, right? Is bringing your knowledge and experience to bear. Okay, so now that we've talked a little bit about diseases, we've covered one, right, influenza. Now we'll talk about a little bit of epidemiology so that when we talk about other diseases, we've got a kind of a clear framework to think about how diseases are tracked, how we know how many people are diseased, right? We know that for some diseases it's important to find the reservoir, okay? The source, right? The chain of infection. Right? How do you determine the chain of infection? That is epidemiology, okay? And so that's why we've got the chain down here, right? And so basically, right, these are the types of things that infectious disease epidemiology is involved in, okay? And so whenever you see a table or a figure that's got yellow highlighted, that is from this particular source, which is in part of your reading. Okay, so first I just want to talk a little bit about data interpretation, right? So whenever you look at data, what are the first things you want to look at? You want to look at the title of it, so you know what the idea is, and make sure you always look at the y-axis and the x-axis, okay? So the y-axis is always going to be something that is measured. That's just how it's done, right? So in this case, this is not about infectious disease, but I like it because what it is doing is showing a clear correlation between the average number of cigarettes somebody smoked per day and the annual lung cancer death rates. And this is the type of information, right? This is from 1951 to 1961, British male doctors, right? This was part of the time when it was being discovered and shown that yes, smoking can lead to cancer, right? And it can kill people. And so this does not show cause and effect, but it is a correlation. Okay, so that's what we're looking at there. And in this particular case, what we're looking at is a table, right, of cholera in the 1850s in London. And what we're seeing right here is the population of Southwark and Lambeth and the number of cholera deaths. And so what was calculated from that is the death rate, which is also called the mortality rate. Okay, and so typically, simply what you do is you take that 844, the number of deaths, and you divide it by the population number. And then, since this is per 1,000, what you do is you multiply that particular number. Okay, so that is the death rate or the mortality rate, which is different than the case fatality rate, which we will go into. Okay, so, and then down here, what we've got is essentially reported rheumatic fever in Denmark in the 1800s. And so what we see is that we've got a steady number, right? Number of what? Well, what we see over here is rheumatic fever cases per 100,000. And so what we see is that it was pretty steady, and then it started declining, right? So how do I use this board? It starts declining over the years. So what is that decline from, right? Well, antibiotics weren't being used until the 1940s, so it wasn't that. It was sanitation, right? So in part of your reading, you'll read about Ignaz Semmelweis, and he actually was a doctor that looked at the number of deaths of mothers from rheumatic fever, okay? And found that there seemed to be a correlation in his mind between the women that were handled and inspected and checked out after doctors had been handling, for example, autopsies as well as other parameters. And so he started talking about hand washing and sanitizing things, okay? And of course, Florence Nightingale really pushed it in nursing, and she's also part of the reading. But Ignaz Semmelweis, just as a quick aside, he ended up dying of the very bacterium that causes rheumatic fever, and he died kind of in obscurity because when he was pushing hard for people to understand that it may actually be the doctor's, bad habits that are resulting in the death of women patients in childbirth, they didn't like that too much. So he was ostracized, and this was before it was shown definitively, right, that bacteria could cause deaths. So this was just something that he saw that correlated. So however, once sanitation became more prevalent, that's why we are seeing in general, because this is just all Denmark, this isn't in maternity wards, what we see is that when sanitation, clean water, washing hands, things like that started happening, then what we see is a dramatic decrease in deaths or in reported rheumatic fever. And so it's a nice historical context. Plus, again, it's showing us how, wherever you're at, some of you probably look at all of these figures and say, I know how to do that, right? But at the same level, this is the 200 level microbiology class, and I need to make sure that you all have those fundamentals. This right here is the first map that was used to look at cases of infection, and this is part of the John Snow case study that you're going to read. You're going to watch some videos, and this was really important in pinpointing certain water that was contaminated by cholera, which led to a lot of deaths. And this right here is showing in graphical form the number of deaths during that particular outbreak of cholera over time, right? From August, you see the number of deaths went up, and then when it went down, okay? And that was after John Snow showed that it was coming from the water and people stopped drinking the water, then the number of deaths went down, right? So that's where epidemiology in this case helped figure out where it was coming from, which is really important, okay? And that's the same figure that I showed you before down there. And so in that case, the reservoir for the John Snow was actually water, right? The source was also the water for the people that got it, but then if they were dying of cholera, the way you die typically is dehydration, and you have massive, horrible diarrhea, that can then cause other people to then ingest it and get sick. So the people that drank the water, the source was the water. Other people that got infected from other people, that was from them. And so the surveillance methods that are used in epidemiology, right, we're not going to go into this in depth, but it involves a combination of, we have people that are getting sick, we need to go in, questionnaires. Some of you may have done this, where you find out, if it was, if they had some type of food illness, what did you eat? Fill out this detailed thing of what you ate, where you got it from, and when you ate it, right? So that's how you deal with that. If it's a sexually transmitted disease, well, you know the partners that you've had, or if you've been injecting drugs, things like that. So for terminology, these are really important. So know these. I think they're pretty self-explanatory, and they're also in your reading, right? So I will just use them. And we've seen this before in the context of influenza, right? And so when we talk about an epidemic, it is essentially a sudden increase in frequency above the expected number. And so if there's an expected number, then that means that that disease is already endemic, right? So that's where when we look at this particular baseline down here, the seasonal baseline is the endemic nature, right? And then when it goes above the epidemic threshold, then we've got ourselves into an epidemic, okay? And so an outbreak is something that happens like all of a sudden there's a measles outbreak, right? It's not, we didn't expect it. Often it's in a location like there's a measles outbreak in a high school or some type of a cult or whatever. And the reason I bring that up is just because, measles vaccination, some people are against these days, and we'll talk about why that's happened and why that's incredibly stupid. All right, that's for later on in the course. And so what we're going to talk about right now is incidence rate, morbidity, prevalence rate, mortality rate, and case fatality rate. Okay, so if we just look at a couple of these diagrams, okay, the one on the left, for example, came from the CDC website, and when we're looking at up above is people living with HIV/AIDS from 1977 to 2005, right? And so what we say is that there was essentially no reported cases, and then we see them, right? For those of you that are my age or older, you remember in the 80s, right? Or a little bit younger when this was first being discovered, right? Very scary times, had no idea what it was. And so it increased, and then it got to the point where treatment, understanding how it spread, so using condoms, and then when we started getting cocktails such as antiretrovirals, things like that, is where we started leveling it off. And then unfortunately, well, not unfortunately, I'll take that back. Then we have an increase, but that is because largely the death rate was going down. So more people are living with HIV/AIDS right now because of the drug cocktails, because of people not being promiscuous if they're infected with it, right? So that's the number living. So down below is the new infections, and that's the thing that we like to see, that this is flattened off, right? And that is for all those reasons that I just talked about. So in this particular case, we saw that it was increasing during the time when it was breaking out, and then it's somewhat stabilized, although the numbers still are not good for new infections in the United States and elsewhere. In this case, look at this, we've got one axis. Look at how the same numbers look on a different representation. Anytime you see numbers on both sides, make sure you look to see what it is and make sure which of these diagrams is for which. Okay, so for incidence, okay, when I'm pointing my finger at this peak, what is the incidence of this? And in this case, we just go over to the incidence right there, and so 80, and it's saying 1,000. Okay? Whereas if we go over in the same year and we look at the prevalence, we go down, right? And we move over this way? Nope. Right? Because we're looking at prevalence now at the blue line, so we don't go over to this one. Now we need to go over to here, and what we're looking at is essentially 100,000. And so the reason to do that is so that for here, you really can't see the makeup and the breakup down. You can see that there's a huge difference, right? But this allows us to maximize both of the axes so we can really see how the incidence went up and it went down. Okay, so what is incidence and what is prevalence? Well, we already see that prevalence essentially is people living with it. That is the total number of people that have it. Incidence represents new cases. Incidence rate is per population numbers. And so when we look at this is from that epidemiology reading. What we see is for incidence and prevalence, we see what it is, what I just said, new versus existing. And then there's the numerator and the denominator and how we use it, right? Incidence is telling us how many people are getting sick now, and does that mean that there's a high chance that you're going to get infected by it? Whereas prevalence just talks about what is how many people are infected at a given time. Okay? So when we look at these particular equations, these are really important, right? So what we're going to do is we're going to go through and we're going to go ahead and do this math on the whiteboard, and then we'll come back to this particular slide set again. So let's go ahead and do a few example calculations, and the data we're going to use is from 2014. And what we're looking at is the number of HIV infections in the United States for people that are 13 years of age or greater. Okay, so the total US population of that age group was approximately 260 million. The number of total infections, that is HIV infections, in 2014 was approximately 1.1 million people. It's a lot of infections. Really a shame. Okay, well, the number of new infections for that year was 37,600. And then the number of deaths that are directly related to HIV/AIDS was 6,721. Okay, so these are the numbers that we're going to work with. Okay, so for that, let's go ahead and look at, for example, prevalence. So for prevalence, what are the numbers that we're going to use? Right? What does prevalence mean? That means the number of, that's right, total infections divided by the total US population. And so that's 1.1 million divided by 260 million. And if you do that calculation, what we're looking at is 0.0042. Okay, so now you don't report numbers like 0.0042. You go ahead and multiply it by the number that gives us a whole number, and then we'll refer to that, that number that we multiplied it by as the per unit. Okay, so in this case, if we want to get to, say, 4.2 for the prevalence, we would need to move the decimal place over 1, 2, 3, which is 1,000, right? So that's 1,000-fold. So we would report this as what? We'd report this as 4.2 individuals per 1,000. Okay, so in that instance, think about it, 4.2 people in 2014 were infected with HIV per 1,000 people in the population. Okay, so let's go ahead on to incidence. And in this case, I use incidence like cumulative incidence, right, instead of just straight up incidence. So for incidence, what we're going to use is, what does incidence mean? Right, that is the number of, what's on the numerator? That's right, the number of new infections. So that case is 37,600. And what do we divide that by? So we're looking at as the number of new infections in the population, right? And so that would be the same number as before, 260 million. And so when we do that division, what we end up with is 0.000145. Okay, so then how are we going to report this? Go ahead and take a guess at it and write it down. Okay, so how many decimal places did you move it? Okay, so again, we want to get to the first whole number, or in this case, we can go to 14 because 14 is just a number that's a lot easier for a lot of people to think of, 14 people instead of 1.45, right? And we could even round that up to 15 people. Okay, so if we do that, it would be 1, 2, 3, 4, so that'd be per 10,000, or we could go 5. Let's go 5 and say that is 14.5 people per 100,000. All right, so in terms of new infections, 14.5 per 100,000, or you could say 1.45 per 10,000. So either way, totally works. Just make sure that all the math works out. Okay, so let's go to mortality. So mortality is what? Mortality is the number of direct deaths, 6,721, divided by, okay, divided by what? That would be the total US population, or would that be the total infections? Would that be the new infections? Which would that be for mortality? Okay, it would be the total US population. So in this case, we're sticking with the same denominator, 260 million. And when you do that number, if we just look at this number, we know that it's going to be smaller. So each one of these numbers in the numerator keeps getting smaller. So even if we just think about checking our own work and making sure that what we're writing makes sense, then we'll use that. Okay, so in this case, what we end up with is for mortality, 0.000026. Okay, let me go ahead and just make a couple of dividers here so we're not getting confused in terms of which one these are. And so that would be, if we move it over, 1, 2, 3, 4, 5. Is that right? 1, 2, 3, 4, 5. Yeah, so equals 2.6, right? And if we moved over by 5 per 100,000. Okay, so things are looking appropriate right now. And so the last one that we're going to look at is case fatality. And case fatality is what? It's the number of direct deaths, right? Divided by what? What are we going to use here? Number of total infections. Okay, so in that case, what we're looking at is 6,721, but finally we have a new denominator, and in this case the total infections is 1.1 million. And so then you can do the math and you can end up with a number that is appropriate. In this case, it ends up being 6.1 per 1,000 cases. So each case is the number of infected people, right, that we know of. And so the direct deaths divided by the number of cases, that's how we get the case fatality number. And so for all of these, just the key things to remember in terms of conceptually, it's a good thing to understand how all this works together, because you don't want to confuse prevalence and incidence or mortality or case fatality. right? And so if you see an equation like this on a quiz or exam, you'll be given numbers, and then you'll be given scenarios where you'll have a variety of different numbers over numbers, numerators and denominators. And so as long as you can do this on paper, you should be fine when it comes to any type of assessment that asks you that. Okay, so now that we've got that sorted out and you know how to do the calculations and you've done a couple of examples, let's look at this incidence rate, the person-time incidence rate, which is the more complicated one, which, compared to cumulative incidence rate, cumulative incidence is often just referred to as incidence. It's a little confusing, I know. Typically, when I refer to incidence, I'm actually referring to cumulative incidence, and you'll see other people do the same thing. But true incidence is this person-time incidence rate. And I just want to go through it just to explain. I'm not going to have you do incidence rate like this. Okay, if I give you an equation to do, it will be of the cumulative incidence rate. But the reason why this matters is this is, person-time incidence rate. And so if you look at this particular situation, we've got 4 categories, or 3 categories: never smoked, ex-smoker, and smoker. The number of each, and then over 8 years, the person years of observation. And so what we see right now is that the 70 cases of stroke in the never smoked, there's 395,000 person years of observation over 8 years. However, for the smokers, where we've got 139 people, there's actually less person years of observation, right? So why is it that we've got almost twice the number of people, and over 8 years there's less person years of observation? That is because the people in this study, when they had the stroke, then they had the stroke. So if the smoker had a stroke 2 years in, then that's only 2 years of person year. Then they had the stroke, right? So if never smoked, a lot of those people never got didn't get stroke until year 7 because they never smoked, then essentially there's going to be more person years involved, right? So that right there, the fact that this is a higher number of people and a smaller number of person years, that means that a lot of smokers got their strokes a lot quicker, right, than non-smokers for the people that got stroke. And so what we see then is that bears out in the incidence rate for stroke. And that is by taking the number of new events in a specified period and dividing it by the number of persons exposed. Okay, so I'm not going to have you do one of these, but I just wanted to make sure that you at least got an idea for why it's important. And what you may see, for example, in nursing, is you may do this for something like the number of people with pneumonia, right? If 200 people in the month of January have pneumonia in the hospital, and only 50 of those, right, if 50 of those people got it while they were in the hospital and 150 people came in, maybe what you're interested in is the number of people that got pneumonia in the hospital. So when they got to the hospital, this person got pneumonia 7 days in, this person got pneumonia 45 days in, you're getting your person years. Okay, so that's a kind of a common way to look at this from a healthcare perspective. And so more data interpretation, again, this is just showing how to interpret data, because you will have to interpret data on quizzes and exams, right? And so basically what we're looking at is the number of people living with AIDS, HIV/AIDS here. And so which axis is that, the left or the right? That's the left, right? And so what we see then is in 1998, there were 30 million people. And if we look at 1998, the prevalence, that is, we then go over here, and that is 1% prevalence, right? So the new infections in that particular year. And so what influences prevalence, right? So prevalence, first of all, what is prevalence? The total number of people that have it at a given time, right? And so that's one of those things where, if you have a disease that somebody has for the rest of their life or until they die from it, right? But with HIV/AIDS, people can live now for decades. So those people are going to live longer, so that's going to mean that there's a larger number of people living that. Whereas other diseases that you get cured from, right, or that kill you, if that's a shorter period of time, then that's going to be less number of people that are part of that, okay? So that's what influences it, the duration of the disease, how many people are migrating in and out. We've got a new drug which cures it or allows you to live longer, all those things, and that's pretty much common sense, okay? So what I would kind of like to bring us out on for the last topic of this particular is this idea of figuring out who infects who, and that is what we're looking at is this cluster diagram, right? And so what we're seeing here is that this particular individual right here infected how many people? That's right, 3. So infected 1, 2, 3, okay? And so each one of those people infected 3 in this hypothetical case, right? And so that is showing how one person infects 3, each of those infect 3, so on and so on. And so diseases can run rampant through populations that are susceptible, right? So if you're not susceptible, for example, in this case, this person right here was not susceptible to the disease, so although they interacted with the person, right, they may have otherwise got infected, but they didn't because they are not susceptible. And in this case, in the book, this diagram is talking about immunity, okay? So since this person was infected, that person did not give it to these 3 people that otherwise that person would have given it to. So this is herd immunity. That is that the increased number of people that are vaccinated for a particular disease, you are protecting the unprotected. And that's why it is so maddening that some people are stopping vaccinate their kids or stopping getting vaccinated from diseases which are cured, because if you get sick, you're going to pass it on to other people, right? And some people, because of other health reasons, they cannot get vaccinated, right? So people that have health conditions that cannot get vaccinated, they're going to be even more susceptible. And so this is a hypothetical example of herd immunity. And what I want to bring up right now is a real live case in a situation where there essentially was no herd immunity, and that is the SARS outbreak and then pandemic of 2003. So for those of you that remember this was crazy. I had I was able to follow this in real time as it was happening because I was a scientist studying this type of stuff, not SARS. But basically, what we're seeing is that, look at this one individual right here. This is patient 1. Look at how many people patient 1 infected. Okay, I'm not going to count that, but basically, maybe I will. No, I won't, but infected this person, infected this person, infected this person, infected this person, etc. Now, some of these people, we don't have evidence that they infected anybody else. Maybe they were quarantined, right? Maybe they died, right, and didn't infect it, or maybe they just weren't around people to get them infected. However, other people, look at this, patient 6 was infected by patient 1, right? And then patient 6 spread it to all these people, which in turn spread it. So you can see that there's these large clusters, because when SARS came out, your average person had no immunity. There's no vaccines. It hadn't broke yet. Now later on, after they did a lot of epidemiology study, very, very cool case study, very interesting, they found that a lot of animal handlers at these open wet markets where people in China are selling animals to slaughter and eat at home, or they're slaughtering them right there and giving you the meat, a lot of those people that were handling it, they had antibodies in their blood against SARS, and so it's believed that the SARS virus was at some level sporadically in people that handled certain types of animals like civet cats that had them but didn't cause an outbreak. And then something occurred where it got out, maybe it mutated a little bit more, and once one person got it, man, look at all the people that this one person is essentially responsible for infecting all of these people through other people, right? And so that shows the reason we know this is from epidemiology. It's from doing all of the antibody tests, from doing all the tests, from going and interviewing all these people that were infected, right, to see who did you have in contact with, okay? And a great movie that shows how you can track this down is the movie Contagion, which is a really good movie from that perspective. And so the people that spread a lot, the small percentage of people that spread it to a large number of people are called super spreaders. And that's another commonality between SARS and AIDS, and that is that for HIV infection, there are some super spreaders where a small percentage of the people are responsible for spreading it to a large number of people. And so from that perspective, I just want to leave it on this, which is on Blackboard, and this is just showing some of the health agencies that we are indebted to, right? Public health is super important. It's, responsibility to the government to protect its citizens, and protecting them from infectious diseases is absolutely, in my opinion, one of the core fundamental responsibilities of the government. So with that said, that's all I've got for epidemiology at this point.